Cancer treatment, including surgery, radiation, medications, and other therapies to cure a cancer, shrink a cancer, or stop the progression of a cancer. Many cancer treatments exist. Depending on your particular situation, one might receive treatment or receive a combination of treatments. Its effectiveness depends entirely on the patient. Hello and welcome to Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and today I'm going to discuss about how structure of antibodies is effective for cancer treatments. Researchers at the University of Southampton have gained unprecedented new insight into the key properties of an antibody needed to fight off cancer. The interdisciplinary study published in Science Immunology revealed how changing the flexibility of the antibody could stimulate a stronger immune response. The findings have enabled the Southampton team to design antibodies to activate important receptors on immune cells to fire them up and deliver more powerful anti-cancer effects. The scientists believe their findings could pave the way to improve antibody drugs that target cancer as well as other autoimmune diseases. In the study, the team investigated antibody drugs targeting the receptor CD40 for cancer treatment. Clinical development has been hampered by a lack of understanding of how to stimulate the receptors to the right level. The problem being that if antibodies are too active, they can become toxic. This latest research harnesses this property of the hinge and explains how it works. The researchers call this process disulfide switching. Researchers analyzed the effect of modifying the hinge and used a combination of biological activity assays, structural biology, and computational chemistry to study how disulfide switching alters antibody structure and activity. The approach was to analyze the structure of the antibody in atomic detail using the method of X-ray crystallography. While the resulting picture is very accurate, the information on how they move their arms is missing, and they needed an image of the antibody in solution, for which they used an X-ray scattering approach called SASS. They then used mathematical models and a chemical computing approach to analyze the data using the Southampton High Performance Computing Cluster IRIDIS, the researchers concluded. They also concluded that the study had given new information about how to engineer antibodies to deliver a better immune response. They proposed that more rigid antibodies enable the receptors to be bound closer together on the cell surface, promoting receptor clustering and stronger signaling for activity paving path for cancer treatment. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.